Hello, my name is Jessica Hartman, and I'm a postdoc in the Meyer lab. Joel Meyer is the leader of Project 3, the Duke Superfund program, and his research is focused on early life exposures, later life effects, specifically in the mitochondrial toxicity. And my research is kind of a subset of that, and it's looking at how lifestyle factors impact toxicity from exposures. And so really, I want to ask the question, if you have an exposed group of people, say you have 100 people who are exposed to the same chemical, the same amount, not all of them are going to get sick from that. In fact, very few, just a handful, will actually get sick. And so in my research, I want to understand who it is that's going to get sick and how can we protect them. And so in lab toxicology experiments, when we're trying to understand how chemical exposures work, we very carefully control everything else about the experiment except for that chemical exposure. So the diet of the animals, the temperature in the room, the light and dark cycles, and even the physical activity of the animals are very tightly controlled. And so when we want to then extrapolate that to what's going on in humans, we do a similar experiment. Just kidding. We actually have to deal with the fact that the humans are not in this carefully controlled environment. In fact, they are having very different diets and physical activity levels, jobs, sleep cycles. And so you can imagine that this is going to impact how people will respond to the exposures. In my research, I think that a lot of those factors are going to specifically impact a person's metabolism. And so in some cases, having a different metabolism might actually be protective. And you can imagine that a healthy diet and more exercise might actually make a person a little bit more hardy against those chemical exposures. On the other hand, there are some metabolic differences that might make a person sicker, and when they're exposed to a chemical, they might be the ones that are actually responding and getting sick. So in the major project that I want to describe today, I'm looking at how exercise is actually quite protective from neurotoxicity. There's a lot of evidence for this in humans. People who exercise and lead an active lifestyle are known to be less likely to develop neurological diseases, even when they have a similar chemical exposure to other people who do get sick. So now I'm trying to understand how it works using the model organism C. elegans. So C. elegans normally live on agar petri dish, and they crawl using this beautiful S-shaped motion that you're seeing in this video here. And so when it comes time to exercise them, a lot of people think that it's going to be something like a worm on a treadmill, something like this. But in reality, all we do is we submerge them in liquid. So when I've taken a drop of liquid and put it on this same worm, instead of making this S shape, the worm actually makes a C-shaped motion. And you can see that it's much more vigorous compared to the crawling motion. So in my research, I've shown that swimming exercise, it does provide a consistent increase in physical activity for those animals, and they are in fact living a more active lifestyle when they're swimming regularly. And so I've shown that the animals who swim regularly, they have a longer lifespan, and they generally age better than their more sedentary counterparts. But I wanted to look a little bit more in the details. So I took advantage of the fact that the worm is completely see-through, so we can light up different cell types inside the worm and visualize them under a microscope. So I'm showing here a strain that has green neurons in the head, and you can see the bright cell bodies at the bottom of these pictures and these projections that go all the way to the nose of the animal. And this picture all the way on the left is a really healthy neuron. So You can see that there's continuous projections with no breaks. And then these pictures beside it have some damage to the neurons. So either breaks in that continuous projection, or in some cases, the projections are missing completely. So this is an example of the kind of damage we can see using these animals. So in the experiment, we take a population of age synchronized animals, and we grow them up and we exercise a group of them for 12 exercise sessions over 6 days. And this is essentially the entire reproductive adulthood of the animal. And then following all those exercise sessions, we do a chemical exposure to a chemical that specifically damages those neurons. And we then look at the neurons under the microscope in the days following that chemical exposure. And what I found is that the animals who have exercised actually do much better. Their neurons are having less of the damage, like the breaks and the missing projections that I showed you in those pictures. And so what we found so far is that, like in people, exercise protected the worm's neurons from these chemical exposures. And so now we're trying to determine what metabolic changes happened during the exercise that provided that protection. And in future studies, we want to determine if those changes also are happening and are protective in people, with the long-term goal of introducing these changes in people to protect them with or even without exercise. So exercise as a pill, perhaps, where we could turn on those same pathways that are turned on in exercise in people that may not be able to exercise. And so I'm actually starting my own group, the Hartman Lab, in October, and you can follow our work at our website, follow us on Twitter, and we're going to be really digging into these lifestyle changes and how they affect chemical exposures.